Hello aspirants, welcome back to our channel Food Tech Online. Hope you all are doing good. So today we'll look at few important multiple choice question answers for the entrance examination for CFTRI, that is Central Food Technological Research Institute. So this will be our set 1. So let us proceed. Okay, FoodTech Online, they're soon going to launch an ebook for all the aspirants, those who want to pursue this MSc in food technology in CFTRI. So this ebook actually it will cover the subjects, those are physics, chemistry, biology, mathematics, microbiology, food chemistry and nutrition, and agriculture and dairy technology and food engineering. So there is a contact number given over here. You can contact over here and get the details about it. So now let us proceed. So question number 1, growth of bacteria or microorganisms, basically it refers to, so options are increase in the size of an individual organism, an increase in the mass of an individual organism, or change in the total population, or an increase in the number of cells. So over here, the growth of a bacteria or microorganisms, basically it refers to an increase in the number of cells. So over here, my correct option will be D, that is an increase in the number of cells rather than an increase in the size or mass of an individual organisms. While we can say the size or the mass of an individual bacterial cell, they might change slightly, but the primary indicator of this microbial growth is the increase in the number of cells. So over here my correct option will be D, that is an increase in the number of cells. Question number 2. Which of the following bacterial species is divided by fragmentation? So options are Bacillus subtilis, Streptococcus faecalis, Rhodopseudomonas acidophila, or Nocardia. So the bacterial species which divides by fragmentation, it is option number D, that is Nocardia species. Now this Nocardia species, they are known to reproduce by fragmentation process. So where what happens in this, the filamentous cell, they breaks into smaller, we can say rod shapes, or it can be cocoidal cells. So each of which can grow into a new organisms. So over here my correct option is D, that is Nocardia species. Question number 3. Rhodopseudomonas acidophila, they reproduce by which of the following methods? So options are binary fission, budding, fragmentation, or sporulation. So this Rhodopseudomonas acidophila, basically they reproduce by the binary fission method. So what happens in this, a single cell, it gets divided into two identical daughter cells. So over here my correct option will be A, that is binary fission. Question number 4. The synthesis of new membrane during the reproduction in gram-positive cells is performed by which of the following organelles? So options are nucleus, mesosomes, endoplasmic reticulum, or cytoplasmic membrane. So these synthesis of a new membrane material during the reproduction in gram-positive cells, these are performed by the mesosomes. So over here my correct option is B. So actually these mesosomes, these are the invaginations of cytoplasmic membrane, so which are found in few gram-positive cells. Now they are involved in various cellular process, which we can say this includes the synthesis of new membrane material during the cell divisions. So over here my correct option is B, that is mesosomes. Next question. So question number 5. Equatorial ridge formation in the cell wall, it takes place in which of the following bacteria? So options are Streptomyces species, Bacillus subtilis, Streptococcus faecalis, or Escherichia coli. So over here my correct option is C, that is Streptococcus faecalis. Now what does the term, that is equatorial ridge formation, exactly means? So it refers to the, I'll write over here, it refers to the localized thickening of the cell wall that occurs at the midpoint of the cell wall of the cell, where we can say the division takes place of the cell, where division division takes place. Even this ridge, it is formed due to the, so this ridge is formed due to inward growth of the cell wall. And we can say, and the membrane at the division septum, and the membrane at division septum. Now, in summary, we can say that this equatorial ridge, it is a critical structure in the cell wall of this Streptococcus faecalis bacteria that facilitates the proper division and the formation of two viable daughter cells. So over here, my correct option is option number C, that is Streptococcus faecalis. Question number 6. When septum formation occurs near the pole of the cell, it results to the formation of daughter cells. These are known as, so these are basically known as mini cells. So over here my correct option is option number C. Now what are these mini cells? So actually these are small, anucleate cells, which are produced when the division septums, they form asymmetrically near one of the pole of the parent cell. So over here my correct option is C, that is mini cells. So question number 7. A bleb or fold-like formation occurs in which of the following bacteria? So options are Pseudomonas acidophila, Bacillus subtilis, E. coli, or Streptococcus faecalis. So over here my correct option is B, that is Bacillus subtilis. Now we know that this Bacillus subtilis, it is a gram positive. It is a gram positive bacterium. Now this can form a bleb or a fold-like membrane. Particularly we can say during the, so it can form a bleb or fold-like membrane. Particularly during this sporulation or under any stress conditions. So during sporulation or stress condition. They basically forms this bleb or a fold-like membrane. Now these structures, these are the part of the cell's we can say adaptation mechanisms, and can be involved in the processes like secretion or cell divisions. So over here my correct option is B, that is Bacillus subtilis. So question number 8, which of the following does not occur during binary fissions in bacteria? So options are cell elongation, cytokinesis, DNA duplication, or spindle formation. So over here my correct option is D, that is spindle formation. Now what is the spindle formation? Actually it is a process which is associated with mitosis or meiosis in the eukaryotic cells. So it is associated with mitosis, meiosis in eukaryotes. Okay, now where these spindle formations, these are involved in the separations of the chromosomes. Now bacteria, we know that these bacteria, these are prokaryotes. These are prokaryotes. So over here they do not undergo this meiosis and mitosis process. So thus this spindle formation will not take place in this bacteria. So over here my correct option will be D, that is spindle formation. Next question number 9. L monocytogenes can be isolated from. So what does this L over here means? It means listeria. Listeria monocytogenes. Monocytogenes. So this Listeria monocytogenes, actually it is a bacterium that can be found in various environments like in milk or in the sewage. So over here my correct option is D. Now it's actually a foodborne pathogen which is capable of causing illness in human beings, particularly those who have a weakened immune systems. So over here correct option is both A and B. They can be isolated from milk as well as from the sewage. Next, question number 10. The diarrheal syndrome and the emetic syndrome are the characteristics of, so options are staphylococcal food poisoning, salmonellosis, purpharingeous food poisoning, or bacillus cereus food poisoning. So over here my correct option will be option number D, that is bacillus cereus food poisoning. Now actually this bacillus cereus, it is a bacteria that can produce two types of syndromes. One is this diarrheal syndrome and the another is this emetic syndrome. What does this emetic means? It's a vomiting syndrome. Now this bacterium, even it can be found in the rice, pastas, or other starchy food, we can say better. Starchy foods that have been cooked and left for some time at the room temperature. So over here my correct option is D, that is bacillus cereus food poisoning. Question number 11. Seafoods and seawater they may contain. So options are Vibrio vulnificus, Streptococcus faecalis, Aeromonas hydrophila, or Vibrio parahemolyticus. Now the seafood and seawater they may contain both the bacteria, that is Vibrio vulnificus as well as Vibrio parahemolyticus. So it is a multiple select questions. It is a MSQ type question basically. So these two bacteria, these are associated with seafood as well as seawater, and even consumption of these in the form of raw or in uncooked food, what happens, seafood contaminated with these bacteria, it can lead to various gastrointestinal illness in the humans and others. So over here correct option is A as well as option number D. Question number 12. A characteristic cysteine loop in the structure of the enterotoxin is seen in the toxins produced by. So options are Salmonella, Staphylococcus, Yersinia, or Bacillus. So over here my correct option will be B, that is Staphylococcus. Now actually they produces an enterotoxin that contains a characteristic cysteine loop in its structure. Now, this enterotoxin is responsible for causing symptoms of this staphylococcal food poisoning when it is we can say this is ingested in the contaminated food. And more specific, we can say Staphylococcus aureus. So over here, correct option is B. Next, question number 13. Question number 13. Campylobacter jejuni has been isolated from, so options are chicken carcasses, turkeys, pork sausages, or all of the above. So this Campylobacter jejuni, it is a bacteria that is commonly associated with the poultries, turkeys, and it is also associated with the meat sausages like this pork sausages. So over here my correct option is option number D, that is all of the above. Now consumption of this undercooked or we can say contaminated poultry, it can cause infections in the human body. So correct option is D, that is all of the above. So question number 14. Traveler's diarrhea is caused due to the toxins which are produced by. So options are Clostridium, Bacillus, Escherichia, or Yersinia. So this traveler disease, it is basically caused due to the toxins which are produced by option number C, that is Escherichia coli, that is E. coli. Now this E. coli strains that can produce the, actually these strains actually they produces the toxins, enterotoxins, or we can more particularly say that they produces, these are enterotoxigenic. So these E. coli, that is ETEC, these are a common cause of this traveler's disease, and these bacteria can contaminate a food or a water in areas which has poor sanitation. So over here correct option is C, that is E. coli. Now Now, question number 15, the syndrome resulting from the ingestion of toxins in a mold contaminated food are. So, his options are aspergillosis, enterotoxicosis, mycotoxicosis, or neurotoxicosis. So, over here, my correct option will be C, that is mycotoxicosis. So, what is this mycotoxicosis? Actually, it refers to the illness caused by the ingestion of toxins produced by the molds, or that is fungi. Now these toxins, these are known as mycotoxins, and even they can contaminate various foods in the mold by this mold growth, which okay. So over here my correct option is C, that is mycotoxicosis. Question number 16. Alimentary toxic aleucia is isolated from the grains which is produced by. So options are Alternaria, Cladosporium, Fusarium, or all of the above. So over here my correct option will be C, that is Fusarium species. So Fusarium species, it is a species of fungi. So it basically produces mycotoxins. And these mycotoxins, these are associated with this ATA. That is alimentary toxic aleucia. So this is associated with this ATA. Now these mycotoxins, we can say example like trichothecens. So the trichothenesis. So these are contaminated by the grains and can cause severe illness in the human beings when it has been ingested. So over here my correct option will be C, that is Fusarium. Question number 17. A toxic substance which is detected in the blue cheese is. So options are lutoskyrene, roquefortin, patulin, or ochratoxin. So over here my correct option will be B, that is roquefortin. Now what is this roquefortin? Actually this roquefortin, it is a mycotoxin. It is a mycotoxin which has been produced by the mold produced by Penicillium roquefortin. So it is produced by this mold species. Now whereas this lutoskyrene, actually it is also a mycotoxin which is produced by fungi like aspergillus or penicillium species, and it is primarily associated with the contaminated stored grains. So it is associated with the stored grains. Now this patulin, so this patulin actually it is also a mycotoxin which is commonly found in the I'll write over here moldy fruits of which is produced by the species like penicilliums or aspergillus species. Next, that is ochratoxin. So this ochratoxin, it is also a mycotoxin which is produced by the molds of this aspergillus or penicillium species, and it can produce be produced found in the contamination of various food products like I'll write over here grains. It is found in the contaminated grains or coffees or wines, etc. So over here my correct option will be which is detected in blue cheese, it is roquefortin. Option number B will be my correct option. Question number 18. Alpha toxins are produced by, so options are A. flavus, A. parasiticus, A. niger, or both A and B. Now this alpha toxin, what is this alpha toxin exactly? It is a toxic compound which is produced by the species of fungi, especially the aspergillus species. Now what exactly those species are? Those are Aspergillus flavus and Aspergillus parasiticus. So over here my correct option will be D, that is both A and B. Now these Aspergillus flavus and as well as this Aspergillus parasiticus species, these fungi can contaminate various agricultural crops, tree nuts, corns. So these are specially grown under the warm and a humid environmental conditions. So over here my correct option will be D, that is both A and. Question number 19. Soft drinks are tested for chemicals when they are contained in a. So options are glass bottle, Tetra Pak, metal container, or none of them. So over here, Acha, you have also seen in the market that these soft drinks, these are basically contained in the metal containers. Example, the aluminium cans. Okay, now these are commonly used for packaging the soft drinks. Now these containers, they undergo testings to ensure that they meet the regulatory standards, and these are safe for storing these beverages or fruit juices. So these testings may include the checks for chemical leachings. So these can include chemical leaching checks. From the container material into the drink. Now even it can ensure that the beverage remains safe for the consumption. So over here my correct option will be C, that is metal container. Next question. Question 20. Samples of suspected perishable foods must be kept cold. So options are during collection of the samples, during analysis, during transit to the laboratory, or there is no need to keep the samples cold. So over here my correct option will be option number C, that is during transit to laboratory. What happens, these perishable food samples, this can be prone to various bacterial growth, or they can even get spoiled if it is not stored properly. Now keeping the samples cold during the transit, it helps to slow down the growth of the bacteria, and even it preserves the integrity of the samples until it is reached to the laboratory for the analysis purpose. That's why over here my correct option will be option number C, that is transit during transit to laboratory. So question number 21, neurological symptoms which is caused by this puffer fish poisonings. These are because of, so options are shellfish toxins, tetradon toxins, both A and B or none of them. So over here my correct option will be B, that is tetradon toxins. Now this tetradon toxins, these are also known as tetrodotoxin. In short we say it as TTX. Now so this is actually a primary toxin which is responsible for this neurological symptoms and which is associated with this puffer fish poisonings. Now this potent neurotoxins, what happens, they basically blocks the sodium channels in nerve cells. What will happen? It will lead to the paralysis or other neurological effects. Whereas this shellfish toxins, it is not associated with the neurological toxins, especially which is associated with the pufferfish poisonings. So over here my correct option will be B, that is tetradon toxins. Question number 22. The predominant symptoms in the lower gastrointestinal tract infection is, so options are fever, abdominal cramp, diarrhea, chills, or malaise. So over here my correct option will be option number B. So this lower gastrointestinal tract infection, typically it manifests with the symptoms such as abdominal cramps and diarrhea. So these symptoms, they are often accompanied by this gastrointestinal disturbances, such as we can say for example the nausea, vomiting, or sometimes fevers. While this fever, chills, and this malaise, it can occur in the infections, but abdominal cramp and this diarrhea, it is a hallmark symptom for this lower gastrointestinal infection, tract infection. So over here correct option will be B, that is abdominal cramp and diarrhea. Question number 23. Aeromonas hydrophila, they causes fishiness in, so option is milk, cheese, butter or cream. So over here correct option is C, that is in butter. So this butter, they may gain such flavor from the absorptions of flavor which is developed in the butter during the microbial growth. So this basically, this fishiness, it is caused by the organism that is Aeromonas hydrophila. Now there are other Easter like flavor. First one is this. Secondly, like Easter's like flavor, which is caused by Pseudomonas fraki. Other like rancid flavors, rancid odor, better to say, caused by these lipase producing organisms. So over here for this fishiness flavor, it is caused in the butter. So correct option is C, that is butter. Question number 24. Vacuum packaged chicken, they contain spoilage organisms in Enterobacter, Pseudomonas, Clostridium or Bacillus. So over here my correct option will be that is Pseudomonas species. Now these Pseudomonas species, these are commonly associated with the spoilage of vacuum packaged meats, including these chickens. So these bacterium, they can thrive in low oxygen environment, such as this vacuum package. So they can cause the off flavors, odors or visual changes in the meat as they grow and they metabolize. Now proper handling is needed, even storage, refrigeration of this vacuum package, these are essential to minimize this risk of this spoilage organisms. So over here my correct option will be option number B, that is Pseudomonas species. Question number 25. Sweet curdling occurs at an early stage of. So options are proteolysis, souring, lipolysis or curdling. So what exactly is this sweet curdling first? So sweet curdling, actually it refers to the separation of curds from milk in the milk without a SAR test. So it's without a SAR test. So this occurs during the initial stage of the. Acha, you comment me down the correct option for this question. At what stage? Now, when the milk proteins such as this casein, it undergoes an enzymatic hydrolysis. So it undergoes an enzymatic hydrolysis. Now this process is even different from the souring, which is involved in the fermentation of lactose to lactic acid bacteria, and it even leads to the formation of lactic acid and a SAR test in the dairy products. So this process is even different. Whereas what is this lipolysis process? We know that this lipolysis basically it leads to the breakdown of the fats and the curdling in the formation of curds. It typically is associated with the coagulation of milk protein, we can say. And so you comment me down the correct option for this question. Next. So with this we have completed 25 few important multiple choice questions for the entrance examination of CFTRI. And if you want to practice more questions, then you can install our app, that is FoodTech online app, which is in the Play Store, and you can practice more questions over here. So if you find any of the doubt regarding any of the questions, then you can comment us down below. And if you want more such videos, then subscribe to our YouTube channel for more such videos. And if you like our video, do share with your friends. So till then, thank you and enjoy learning.